Zoominar series. I'm Nikki Evans. I'm the Assistant Director at Sedgwick Reserve. And as many of you know, Sedgwick Reserve is part of the UC Natural Reserve System. We're an open air laboratory for researchers, for students, and we also do public programs. I am excited to announce that this upcoming fiscal year, we'll be able to start doing public programs again. So that includes our walking ecology, our guided hikes, our annual barn dance, which I have not been able to attend yet, but I hear it's just a lot of fun, and other events. So to keep informed on that, do sign up for our newsletter if you're not already on it by going to our website. And you can also follow us on Instagram or on Facebook. So a couple of housekeeping items for our webinar today. There will be a chance at the end for some questions. And if you think of any along the way, feel free to type them into the Q&A box located at the bottom of your screen or into the chat box, and then we'll address those at the end of Kenji's talk. So I'd like to introduce Kenji Hayashi. Kenji is a PhD student in the Department of Ecology and Evolutionary Biology at UCLA. He works in Dr. Nathan Kraft's Plant Community Ecology Lab. And he's also a recipient of the Matthias Student Research Grant Award. And that is a grant that goes to promising students to undertake research in the UC Natural Reserves. So without further ado, I'd like to introduce Kenji, thank him for joining us today. And we're all really excited to learn about what he's been learning out at Sedgwick. Take it away.
Great, awesome. Thanks, Nikki, for that introduction. So hello everyone again. As per the introduction, my name is Kenji Hayashi. I'm a PhD student in the Department of Ecology and Evolutionary Biology at UCLA. And I'm really excited to be talking to you today about my ongoing research at Sedgwick Reserve. Let me just get started here by sharing my screen. Okay, is that good for everybody?
That looks good.
Great, thank you. Okay, again, hello everyone, and welcome to my talk. I'm really excited to be sharing this research with all of you today. And what I'm going to be talking about today is the research that I've been doing at Sedgwick for the past year and a half, where I'm aiming to understand the distributions of annual plant species in this California grassland, which is a really unique and beautiful place. Before I even get started, though, I do want to start off with some acknowledgements, because none of this work would have really been possible without all the support from all these incredible people listed here. And I especially want to thank my advisor, Dr. Nathan Kraft, my lab mates in the Kraft Lab, and also especially staff at the Reserve, Kate and Nikki in particular, who have really been integral to the continuation of my research at the Reserve during these really challenging times. And I should also mention that funding for the research I'm about to talk about is provided by both UCLA as well as the UC Natural Reserve System's Matthias Graduate Student Research Grant, and also some grant from my advisor's grant from the National Science Foundation as well. I also, as a researcher working at Sedgwick, I also want to acknowledge that the land I'm working on is the traditional, ancestral, and also unceded land of the Chumash peoples. And if you are interested in this, I would highly recommend checking out this website linked here, which maps out Native American territories. And you can find out more about this history in historical documents. You can go online, and the Reserve also has some information about this history as well that I would recommend checking out. Okay, so with that, I think we can get started. So as the title of this talk suggests, the research that I'm going to be presenting today concerns species distributions, or in other words, where species occur in nature. Now, this question of where species occur and why is one of the most long-standing questions in plant ecology. And some of the earliest work by Western scientists on this topic dates back to the early 1800s, when Alexander von Humboldt documented the distributions of vegetation across this mountain in the equatorial Andes in relation to the climatic gradients observed there. And I think that this is a really interesting and complex field of study that many ecologists have dedicated their entire careers to. And I would argue that this issue of understanding where species occur and why is becoming even more relevant now, as natural habitats are increasingly threatened by things like climate change and habitat destruction. So for example, by understanding the relationship between environmental conditions and the occurrences of species, we can use statistical methods called species distribution models to predict the current or even future distributions of plant species. And in addition to that, understanding why species presently occur where they do, and conversely, why they don't occur in certain places, can also be really relevant for things like local conservation and restoration of rare or charismatic plant species that we might care about, some of which I've shown here. Now, I believe I mentioned just a moment ago that understanding species distributions can be a really complicated thing. And one of the reasons for this is because species distributional patterns can be observed in really various different ways. And I'm going to illustrate briefly what I mean by that. So taking the Stinia californica, the California goldfields, as an example, at the scale of this single patch shown here that you might even encounter just walking around the reserve, we can see that there are actually points here where the species is present, which basically is where seeds landed, germinated, and then survived until adulthood. And then we can also see that there are points where individuals of this species are absent, such as where there's bare ground here and also where the sponge grass is growing. Now, if we zoom out a little bit though, and observe this landscape as a whole, we can actually start to notice that there are certain areas in this landscape where Listeria occurs in patches, and also there are areas where Listeria doesn't occur at all. And we can even take this even further and zoom out to, let's say, the regional or continental scale, where we can start to map out the entire geographic range of Listeria to illustrate where, geographically speaking, the species can or cannot be found. So in light of this, one of the unifying themes of my research and what I'm going to be talking about today is trying to understand what ecological processes shape species distributions and also entire plant communities across these different scales of organization. Which then brings me to this really interesting question of what controls species distributions? Another reason that understanding this topic of species distributions can be really complicated is because there are actually a wide variety of factors that can go into shaping observed distributional patterns. And in general, though, there are two major types of factors that we commonly think about as influencing species distributions. The first one, on one hand, we have abiotic factors, which are aspects of the physical environment. And on the other hand, we have biotic factors, which are essentially living parts of the environment. Some relevant abiotic factors that can constrain species distributions include things like climate, including temperature and precipitation, topography, habitat type, soil conditions, and in places like California, where fires are an important determinant of species occurrences, fires can also be important as well. As an example, for example, you can imagine that climate is going to be quite different between a coniferous forest in the northern US or Canada and in an oak woodland in California. And in most cases, each of these habitat types is going to contain very different species that are adapted to these very different climatic conditions. So that is one way in which this abiotic environment, in this case climate, could be controlling species occurrences. On the other hand, there are also various biotic factors that can constrain where species can occur. And a particularly important one, which is going to be the focus of much of this talk, is competition for resources among nearby plants. And what I mean by this is that most plant species, for example, shown here, need to draw on the same limited supply of resources like water and nutrients. And this necessarily creates competition for these resources among neighboring plants. Now, you might imagine that a strong competitor might be able to persist in a variety of environments in the presence of these neighboring plants, but a weak competitor might easily be excluded from a locality by these superior competitors, and thus have its distribution truncated for this reason. Some other biotic factors that could limit species distributions include things like the presence of predators, disease, parasites, or even the absence of pollinators that are necessary for successful reproduction. But I'm not going to be talking about most of these today. Just to continue with some background information, interestingly, it's generally thought that the relative importance of these various factors that I just described for shaping species distributions varies depending on the spatial scale under consideration. And what I mean by this is that, if you might recall, I introduced some examples of these different scales of organization that we can study plant communities at. So for example, we have the patch scale, we have this landscape here, and then we have this entire regional or continental scale that we could also study. These can be thought of as these different spatial scales of interest. And in line with that example that I gave about coniferous forests versus oak woodlands, climate is thought to play a particularly important role in shaping species distributions at these very large spatial scales, like this continental or regional scale. Now, conversely, these other factors like species interactions, including competition for resources, are thought to be important at somewhat smaller to perhaps intermediate spatial scales. And things like habitat type and soil, these might also be important at somewhat intermediate scales. Now, you might notice that I'm talking about this sort of vaguely, and this is because it is a little bit vague and it's really hard to study this stuff. And in particular, the interplay of these various factors is quite difficult to disentangle in natural settings. And in particular, I would say that there is an especially large knowledge gap surrounding the role of species interactions, such as competition, in shaping species distributions as we observe them currently. Now, one nice study that starts to get at the role of species interactions in this is this study by Bullock and colleagues, in which they studied the distributions of these two Ulex species. Here, we can see that these two species, shown as red and blue points on this map, are basically seen to co-occur in the same geographic region. However, the occurrence of the two species are separated in space, as you can see here. And from this pattern, it is observed, or it is inferred, that these species are competitively excluding one another within each of their suitable habitat zones. However, I would say that it's still pretty difficult to disentangle the relative importance of abiotic versus biotic factors from observational studies like this one alone. Okay, so having introduced all of that background information, I now want to take you back to this earlier example of the distribution of Lysthenia across spatial scales. The main question that I'm going to be addressing in the remainder of this talk is this question of how do species interactions with the abiotic and biotic environment collectively shape species distributions at the landscape scale, as indicated by this arrow I've drawn here. Now, in order to answer this question, we do need to develop a more precise way to quantify how the performance and therefore the distributions of species is limited by the abiotic environment or the biotic environment. One way to do this is to employ a demographic perspective on species distributions. And what I mean by this is that for a species to be able to occur in a given locality, let's say somewhere in Sedgwick Reserve, it needs to maintain a healthy population of individuals there. And this population persistence, in turn, requires a series of successful demographic events. In the case of annual plants that I study, this means that seeds need to germinate, or actually seeds need to survive in the soil, then germinate. And then those germinated individuals need to produce enough seed to sustain the population going forward. Now, interestingly enough, several of these demographic rates could potentially be influenced by biotic interactions, including competition or predation, things like this. To be more precise though, these demographic rates actually combine to determine the total growth of the population as a whole. And a species must exhibit a positive population growth rate in order to persist in a locality. Or in other words, the population has to be growing and not declining for a species to be able to persist in a locality in the long term. For example, let's consider this hypothetical two-species community. If we start off with this red species at low abundance, but it's able to grow and reproduce over time like this, this indicates that the population growth rate for this red species is going to be positive, meaning that the species is expected to persist in this environment in the long run. Conversely, if the red species is unable to grow or reproduce from low abundance, like this, and then eventually dies off, then the population growth rate is going to be negative, meaning that the species is not expected to persist in this environment. So under this framework, by measuring key demographic rates in different environments, and then quantifying how they're modified by these biotic interactions like competition, it actually becomes possible to separate out the role of biotic versus abiotic factors in shaping the performance of species, which in theory should then help us explain observed distributional patterns that we observe in these landscapes. So this now brings me to my specific research questions. The first of which is this question: how do demographic rates vary in these different abiotic environments that we encounter on the landscape? In relation to that, the next question is, how do competitors modify said demographic rates? And the third question is basically asking, how do these abiotic and biotic factors combine to shape the distributions of these species that we see across these entire landscapes? So in order to address these research questions, I conducted a field experiment at Sedgwick Reserve during the 2019 through 2020 growing season, so a year ago. My study area shown in this picture here is located in this northwestern corner of the reserve, which is pretty far from the base here. And I also want to note that in this picture, there's a cage shown here, but you can just ignore it for now because I'm not using it in this study, and I'm actually just studying the surrounding landscape. Now, I suspect some of you, or perhaps many of you, are quite familiar with the natural history of this reserve, but I do want to provide some basic background information that's relevant for my study. First of all, this reserve has a Mediterranean climate, which means that it has really hot and dry summers and cool and wet winters. So many of the plant species that exist here are these winter annual plants, which are also the ones that I'm studying. This reserve, especially this portion of it, also has a very interesting and spatially variable landscape, where we have these serpentine hummocks over here interspersed among this matrix of grassland. Now, from an experimental standpoint, many of the species here are these small annual plants, which actually makes them ideal for experimentally manipulating, which is why I am using them for my studies. And this is also a very good example of one of these highly invaded California grasslands, which makes the results of this research potentially relevant for things like local conservation or restoration efforts. So that's something I have in mind as well. Okay, so taking a closer look at the landscape in this study area, there are actually two major habitat types that I'm going to be considering in this study. First, over here, we have these serpentine hummocks, which really have these quite sandy and shallow soils. And they contain many of the small native forbs like Vestinia, for example. On the other hand, over here, we have this grassland matrix, which has much deeper clay soils and is presently very heavily invaded by invasive European grasses, such as Avena and Bromus. Now, throughout the rest of this talk, I'm going to be referring to these two habitat types as the hummocks and the grasslands, respectively. And again, the question that I'm going to be addressing is, how is the performance or persistence of species impacted by these different abiotic and biotic components of the environment to collectively shape species currently observed spatial patterns on this landscape? So in order to answer this question, though, we also need to establish a deeper understanding of the life cycle of these annual plants. And illustrated here is what we believe to be a reasonable representation of this life cycle. What this is showing is basically that for these annual plants, each year, starting with a certain number of seeds, a certain fraction of those are going to germinate and then produce these little small seedlings. These germinated individuals will then mature and then go on to produce more seeds that will then enter the seed bank. Of these seeds that didn't germinate in this year, a certain fraction of those are going to survive until the next year. And together, the population will be growing if the final number of seeds is larger than the initial number of seeds here. This life cycle diagram can then be translated into a very simple mathematical expression, which basically says that the population growth rate of a species is a function of its germination rate here, its fecundity, which is seed production, seed survival in the seed bank, and also some of these terms may be modified by competition. What this means is that by measuring these three demographic rates and the effect of competition on them, we are actually able to calculate species population growth rates pretty easily. For those who are interested, here's the full mathematical expression for this model. I'm not going to take the time to explain this in depth here, but you can see that each of the terms that I just mentioned in the previous slides, including germination rate, seed survival, fecundity, and competition, these all reside somewhere within this population model, and that allows me to calculate population growth rates from my experimental measurements. I'm happy to answer any questions about this model later if you have any. So for my field experiment, which I'm going to explain in a moment, I chose to study these 8 different native annual plants that represent a diverse array of plant species found in this general area. These focal species are listed here and include acmespon, shown here, Chenactis glabrioscula, Festuca microstachys, which is a grass, Hemizonia, which is tarweed, Lasthenia, goldfields, Plantago erecta, which is the California plantain, Salvia, which is chia, for those of you who enjoy eating chia seeds, and then we have Europappus, or silver puffs. For simplicity, for a lot of this talk, I'm going to be focusing mostly on contrasting results for Festuca, the only grass, and Plantago as the representative forb, because these two species exhibit some interesting and sometimes contrasting patterns. Okay, so with the aim of experimentally quantifying those previously mentioned demographic rates, what I did is I planted and grew individuals of these 8 focal species in these 7 environmentally heterogeneous study sites distributed across this focal landscape. 3 of these sites, shown in blue, were located within the grassland habitat, while 4 of these sites were located on the hummocks. At each of these study sites, the focal species were grown under one of 2 different treatments. The first is growing them in the presence of natural competitors that naturally exist at that site, shown on the left here. And the other treatment is growing these species without natural competitors. And what I mean by that is I manually removed all neighboring species and individuals so that my focal individuals of my focal species will be growing alone without interacting with neighboring plants. So for each site and each treatment and each species, I then tracked the germination and fecundity of approximately 10 focal individuals throughout the entire growing season. In terms of what this actually entailed, basically what I did was for each experimental unit that I had, I planted a certain number of seeds in a given point, came back to them once they germinated and counted the number of seeds that had germinated, something like this. And then I thinned this patch down to a single individual, which I then monitored that individual throughout the entire growing season And at the end of it, I measured this individual's fecundity by counting the total number of seeds that it produced at the end of its lifespan. So having 7 sites, 2 treatments, 8 species, and then 10 focal individuals per species actually meant that I ended up tracking over 1,000 individuals in total during this growing season last year, which was a lot of work. And it didn't help that this happened during some COVID times as well, but I got through it. So this experimental setup basically allowed me to answer my research questions, which as a reminder are basically asking how the abiotic environment, biotic environment, and how these modify species demographic rates and therefore population growth rates, and how this influences species distributions. What I'm going to do now is I'm going to present the results of 3 parallel analyses evaluating how germination rate, fecundity, and then the overall population growth rate varied by habitat type and experimental treatment, which is basically the presence or absence of natural competitors that exist at each of these study sites. Okay, so starting off with germination. This figure here shows basically how Plantego's germination rate, plotted on the y-axis here, varies by habitat type, plotted on the x-axis, either as a hummock or a grassland. And the color coding here is representing whether or not these values were obtained with or without the presence of neighbors. Green means that the neighbors were present, whereas orange means that neighbors were removed in the treatment. So what we can see here is that in both the hummock and the grassland habitat, the germination rate for Plantego appeared to be higher when neighbors were present, and this is consistent across both habitat types. We can actually contrast this pattern to what we see for Festuca, the grass, which actually exhibits a higher germination rate when neighbors were removed compared to when neighbors were present. And again, this effect seems to be consistent across habitat types. So the first impression here is that there seem to be some species-specific responses going on here with respect to the habitat and neighbors when it comes to germination rate. Now, we can summarize these demographic responses to habitat type and treatment for all of these study species in this figure shown here. And what is being plotted here is basically we are contrasting germination rate with or without neighbors. So with neighbors and without neighbors in these 2 different habitat types, which are the hummocks and the grasslands. And this figure is a little bit messy, so I would appreciate it if you could bear with me for a moment. But some of the patterns that we could observe here are that first, the effect of neighbors on germination does seem to be somewhat species specific in this case. Plantego, which is this light green species, and perhaps Acmaspon are the only species that really notably had less germination when neighbors were removed, meaning that all the other species basically benefited in terms of germination by removing neighbors. And the third thing that we can see here is that it's a little bit hard to discern, but the negative effects of neighbors on germination are sometimes much more pronounced in the grassland habitat than in the hummock. And what I mean by this is that in the absence, compared to in the absence of neighbors, when neighbors are present, there's a dramatic decrease in germination rate for some of these species, which is much more significant than the decrease in germination rate due to neighbors for if you are growing in the hummock habitat. Now that was a little bit messy, but these patterns become even more clear when we basically average across all these species. And the results shown in this graph, basically you can think of this as some generic or average annual plant as represented by my study species and how they're responding to these habitat type and neighborhood conditions. So what this is showing is that in the absence of neighbors, germination appears to be about the same in both habitats in general, but when neighbors are present over here, germination rate is reduced more by competitors in the grassland habitat than is the case in the hummock habitat. And this pattern suggests quite a strong or a stronger role of competitive interactions in limiting the occurrences of species in the grassland habitat, with the caveat that this is still only one of the demographic rates of interest. Now, I do want to note, however, that the germination rates I'm talking about here are likely some combination of actual germination success as well as early season survival, because there's a slim possibility that some of the seeds germinated and then the seedlings died before I was able to count them in the experimental plots, just due to the timing of things. However, I don't expect this to really have a large bearing on the main conclusions being drawn here. Okay, so we can now move on to the results for fecundity, where we can basically repeat the same set of analyses and ask similar questions. Here, we are examining how fecundity of Plantago plotted on the y-axis varies by habitat type and also experimental treatment, which is the presence or absence of neighbors. What we see here is that in both habitat types, fecundity is much higher when neighbors are absent or removed, meaning that if you're going alone, you produce more seeds. However, the reduction in fecundity that arises from the presence of neighbors or having to compete against other individuals is much greater in the grassland habitat than in the hummock habitat, as can be seen in this difference in these two boxes here. Just to give you sort of a sense of what's going on here, these two photos illustrate some of the variation in fecundity that can arise under these different conditions. So if we compare the size of a mature Plantago growing in the hummock versus in the grassland habitat, both without neighbors, we can see that the biomass and therefore fecundity of the Plantago growing in the grassland is so much higher than in the hummock habitat. And furthermore, if you look at this graph or the results in this graph, what this tells us is that the difference in size and therefore seed production of this species when neighbors are present versus absent in the grassland is going to be an even larger difference than this difference illustrated between the two habitat types in the absence of neighbors. So this effect of neighbors for reducing fecundity is really large in these grassland habitats. And it still exists in the hummocks, but perhaps not as strongly. Now, unlike in the case of germination, where Plantago and Festuca displayed sort of opposing patterns, in this case, Festuca actually exhibits the same sort of pattern that Plantago does, where fecundity is reduced by neighbors, but more so in grasslands than in the hummock habitat. In fact, if we look at all of the species, this pattern is almost uniformly shared across all of the study species in my experiment, as shown in this graph. That is, fecundity is generally higher when neighbors are removed or absent, as opposed to when neighbors are present. However, the effect of competitors for reducing fecundity is much more stark in the case of the grassland habitats compared to in the hummocks. Similarly, when we average across all these species and look at some generic or average species, what we see is that there appears to be a similar fecundity between hummock and grassland habitats when natural competitors are present. Excuse me, but a release from competitors is much more beneficial in the grassland habitat compared to in the hummocks. Again, similar to what I talked about previously, this suggests that competitors play a really large role in limiting the performance of plant species, especially in these grassland habitats that aren't the hummocks. Okay, so having analyzed germination and now fecundity, which are the focal demographic rates that we're interested in, we can now move on to analyzing or considering population growth rates as a whole, which are what ultimately matter for population persistence rate. So the population growth rates shown here, as I mentioned earlier, are calculated using the experimentally estimated demographic rates that we just talked about, and also by using that equation for the annual plant life cycle that was shown in one of the previous slides. So here, we are examining the population growth rate of Plantago with respect to habitat type and experimental treatment. Now, the first thing we can see is that Plantago is actually predicted to persist in both habitat types, with or without competitors, except in the natural grassland community here, where there is uncertainty overlapping a 0 to negative population growth rate, meaning the species may or may not persist. I guess to rephrase what I just said, the population growth rate of this species, Plantago, is expected to be positive, meaning the population is going to be growing in the hummock, regardless of what your competitive environment is, and in the matrix, if your competitors are removed. But when your competitors are present there, it's more questionable whether or not you are going to survive there, and most likely you probably won't. Is one of the potential outcomes here. Now, what's really interesting here is that this is very qualitative, but this is a species that is actually largely constrained to these hummock habitats, and yet it achieves its highest population growth rate in the grassland habitat if we're able to grow there alone in the absence of these natural competitors. The overall pattern here, though, is similar to the case of fecundity that we just talked about, in that population growth rate is higher when competitors are removed, but this effect of competitors and population growth rate is much more significant in the grassland habitat than in the hummock habitat. Again, this pattern is overall similar between Festuca and Plantago, except in the case of Festuca, which is a grass, it's actually expected to survive relatively decently in the natural grassland habitat among its competitors. So again, if we take a look at all of the study species, this pattern that I just described is almost uniformly present across all of these species as shown here. That is, population growth rate is higher when neighbors are removed as opposed to when neighbors are present. So it's higher when neighbors are removed or absent, and it's lower when neighbors are present. However, as is the case with the previous slides, this effect of competitors or neighbors in reducing population growth is much more significant in this grassland habitat than in the hummocks. Now, really interestingly, there are a number of species that appear to be unlikely to persist in the natural grassland environment due to having a negative or near 0 population growth rate. So these species include Lasthenia in yellow, Salvia in purple, Chenactis in this little salmon color, and then also potentially Plantago, depending on the uncertainty in our estimates. Now, notably, most of these species, if not all of them, are species whose present distributions are, again, largely constrained to these hummock habitats. And the fact that these species are expected to generally perform well in grasslands when the natural competitors are removed, perhaps with the exception of Chenactis, means that these competitors in the grasslands probably play a really major role in limiting the distributions of many of these native forbs, which might be why we don't see some of these native forbs in this grassland habitat. Finally, for this final set of analyses, we can, again, average across these species and examine some generic or average plant species in our experimental context. And we can conclude from this figure, again, that competitors in the grassland habitat, shown in blue here, generally play a much larger role in limiting the population growth rate of these species compared to in the hummocks. Okay. Now, just to summarize all of that, all the insights gained from this experiment. First, it seems pretty safe to say that the germination rates and fecundity of these species do vary spatially by habitat type, although the degree to which this is the case seems to depend on which species we're talking about. Second, it seems to be rather clear that competitors just generally negatively impact the performance of species, as might be expected. But this effect is much more pronounced in the grassland habitat than in the hummock habitats. Finally, third, these results do appear to suggest that natural competitors are strongly limiting many native forb species from occurring within these grassland habitats. However, this last conclusion here, as I mentioned earlier, is somewhat qualitative, based on qualitative observations, and probably needs some more rigorous testing. So the next step, the immediate next step for this research is therefore to more rigorously compare species' predicted performances with and without competitors to their observed distributional patterns on this entire landscape that I'm studying. Now, in order to do this, the first thing we have to do is to establish these relationships, ideally a continuous relationship between certain environmental variables, such as soil depth, and also population growth rates. So although I focused on talking about this binary habitat types, which are the hummocks and the grasslands for the majority of this talk, in reality, there's also environmental variation within those habitat types as well. So focusing on certain explicit environmental variables might actually yield more precise results for us. And so based on the results that I was just talking about, this relationship between, let's say, soil depth, which varies across this hummock grassland gradient, and population growth rate might look something like this. This is not confirmed. This is one idea or one hypothesis, if you may. But once this sort of relationship is established, what I can do is we can couple it with a map of environmental conditions to project population growth or population performance across this entire landscape. And this will then allow me to compare predicted performances, either with or without competitors, to the actually observed occurrences or abundances of these species at each of these locations on the landscape. And then I can ask, to what extent are species distributions potentially limited by the abiotic environment or responses to competitive environment? So in order to do this, I am currently actually in the process of quantifying the properties of the soil environment and surveying plant community composition at these approximately 70 spatially distributed locations across this landscape. This was actually work that I had initially planned to do last year in tandem with my field experiment, but unfortunately that didn't happen due to COVID. So I'm doing that right now and hopefully wrapping up this effort soon. Okay, so moving on to another future direction. My field experiment has basically allowed me to quantify whether or not competitive interactions are influencing species performances and potentially distributions. Now, for some of you who are familiar with California grasslands or familiar with the ecology of Sedgwick Reserve, the conclusion that the effect of competitors is particularly strong in the grassland habitat may not be entirely surprising, given that these grasslands are generally dominated by these European invasive grasses, like these. And in turn, the serpentine hummocks are often thought to serve as refugia for many native forbs. So in a sense, it could be said, I guess, that my experiment has confirmed this understanding of the spatial patterns on this landscape through a very detailed look at the demography and population growth of these different species. However, there still remains this question of what is the relative importance of these demographic factors, germination, fecundity, competition of these species for shaping landscape scale population growth. And in order to answer this question, what I can do is I can employ an approach called variation partitioning, which basically allows me to ask what % of overall population growth comes from variation in each of these demographic factors. How much of it is due to competition? How much of it is due to fecundity and germination? This can help me elucidate which of these factors is actually one of the most important things in controlling species performances and therefore distributions. As a secondary question to this, I can also ask how this relative importance of these different demographic factors varies across spatial scales. So this goes back to this question or this topic about how species distributions might be controlled differently at different scales. For example, as defined by these different red outlines on this map. For example, one question I could ask is it actually true that the relative importance of competition diminishes towards these larger spatial scales, as is often assumed or suggested in much of the literature? Understanding this sort of scale dependence can serve as a bridge between the work that I'm doing, basically in this tiny corner of Sedgwick Reserve, and other research that aims to understand species distributions at broader spatial scales. And the final thing that I want to talk about is this third next step, which stems from one of the limitations of my study in its current form, which is that some of these results, as I mentioned earlier, are somewhat species specific. Therefore, it's really hard to be general about these results. Therefore, as a third next step, I will be asking whether plant functional traits, which basically represent aspects of a plant's physiology and morphology, can help predict species responses to competition exerted by these natural competitors. Now, functional traits, these morphological and physiological characteristics, these can include aspects of leaves, as shown here. It could also have to do with roots or even whole plant structure. And the utility of this approach is that by changing our focus from species identities, like let's say like acmospan and asthenia, we can change that to look at what are the physiological and morphological characteristics about the species that might be causing it to have these responses to the environment and to competition. And by doing this, we might be able to achieve a slightly more general understanding of competition and the species distributions from this experiment. I think it's worth mentioning, I guess, that previous work at this field site has actually successfully linked some of these functional traits to species performances and competitive dynamics. So I think that this might be a potentially fruitful direction to go in. And actually, some version of this work is currently being spearheaded by Tim Foster, who's an undergrad, an amazing undergrad in our lab. And I'm really excited to be working on this with him. Okay, so now I'm going to conclude this talk by summarizing some of the key takeaways from this entire presentation. The first is that the results of my field experiment show how the performance of these plant species that I studied in this annual grassland of Sedgwick Reserve seem to be determined by this interaction between the abiotic environment, such as soils, and the biotic environment, such as competition, which means that in order to understand where species occur on this landscape, you need to look at both of these. You can't just focus on one of them. Perhaps the most important finding from this research so far, though, is that there appears to be a relatively strong support for the role of competition in shaping species distributions at the landscape scale, especially in this grassland habitat that I've talked about a couple of times now. And I really believe that the detailed demographic understanding of species performances and therefore their resultant distributions obtained in this study might have potential to be applied towards local conservation and restoration efforts, and perhaps even applied towards more accurately predicting species distributions under climate change in the future as these methods advance. Finally, the ongoing and upcoming work that I'm conducting at this reserve really aims to extend this work that I just talked about today to provide a more detailed understanding of the interplay between these abiotic and biotic factors in shaping species distributions across these different spatial scales that I'm really interested in. And with that, that'll be the end of my talk. So thank you very much, everyone, for listening.
Thank you so much, Kenji. That was wonderful. You did a really great job of taking this really complex topic and breaking it down. I found that really fascinating. So if you're listening and you have questions, do type them into the Q&A. So Kenji, our first question comes from Dennis Beebe. He asked, does your study shed any light on what native annual plants might have dominated the grasslands prior to invasive European grasses?
That's a really interesting question. So I can actually pull up some results that might shed some light on this. So I'm going to go back to one of the slides I showed earlier. So here is the graph that basically illustrates how the population growth, which is ultimately what we care about, of these different species depends on both the presence and absence of neighbors, and also on what habitat you're growing in. So the complicated thing is that we know from, well, I believe that many studies have suggested that abundances don't necessarily correlate strongly with things like population growth. So that's a complicating factor. However, one thing that we can see is that one of the species that achieves the highest population growth in the grassland habitat, if there were no grasses there, one of these is Hemizonia. And for those of you who are familiar, this is tarweed, and tarweed actually exists among these grasses. And it's one of these few annuals that does a really good job persisting, because perhaps it can grow really tall as well. So my prediction, based on just walking around this reserve and also some of these results, would suggest that the tarweed is going to be one of these that might be really dominant if all these grasses were removed. However, it's really hard to predict that just from these results, because honestly, any of these could probably occupy a fair amount of space once the grasses are removed.
All right. Well, thanks for that. Yeah, I had a couple of questions while you were showing some of your results. You said there were a few plants that had higher germination rates when neighbors were present. Why might that be?
So that's a very puzzling result that, to be honest, I'm not entirely sure yet either. Let's see. Yeah, so there are some species here that basically seem to, for some reason, have a higher germination rate as quantified in my study when their neighbors are present. This could be something real, biologically speaking. It's also foreseeable, although unlikely in this case, that this has to do with measurement error, but I don't think that's probably the case, at least for Pentago. So I guess, unfortunately, my answer to that question is that I'm not entirely sure. I haven't read up on why this might be the case, but it's sort of an interesting thing.
Yeah, well, good research often brings up a lot more questions to be asked. Gloria and Dale Murray suggested, could it be that neighbors provide shade and therefore more moisture?
In the context of germination,
yeah.
Or just in general? Yeah, so definitely one of the things that you can observe, especially in the grassy habitats, is that neighbors do provide a lot of shade, and this retains a lot of moisture. However, in the case of most of these species, what I've noticed is that more so than the amount of additional moisture, the limitation, the limited access to light seems to be much more detrimental at this really small seedling stage. So I suppose that's one possible explanation for why we might see this negative pattern here. But at the end of the day, I'm not entirely sure about that.
Okay. We had another question from Karen Talene Lawton. She was asking if you could, to be clear about competition, does that just mean any plants, same or different species, spatially close by? Or does it mean specific plants that are known to be detrimental to growth of the target species?
Okay, yeah, so in this particular experiment, we are being agnostic, agnostic about what surrounding plants we're talking about. So this effect on germination, this effect on fecundity, this is the net effect of whatever surrounding species have on these individuals that I was tracking. So the question there is really interesting because it's possible that there are some species that don't really impact one another or may even benefit one another in certain circumstances. But if there is a negative, let's say a negative effect of neighbors, that means that overall, with everything around it, this was detrimental to the individuals I was tracking.
Yeah, you had mentioned that you had, so you had just the natural existing species around for the neighbors. So that included the invasive European grasses.
Yes.
Okay, great.
Yeah, because what I wanted to understand was why do we see the patterns that we currently see right now, which is dependent on these resident communities in their natural state.
Okay. We had another question from Dolly and Charles Mullen. They asked, are there also different determining factors in the hummock versus grassland habitats for how much competition there typically is? For example, do deer eat more forbs in one area versus another?
Oh, that's really interesting. So I think there are a couple of things. For example, in terms of competition for certain resources like water, water, I believe, becomes much more limiting, much more earlier in these hummocks because they just have a lot of drainage. They don't retain water very well. The soil is very shallow. So in that sense, what they are competing for and when they are competing for it is going to be slightly different. In terms of removal of potential competitors through things like predation or let's say eating forbs, these deer eating forbs, I have noticed that this appears to be more common in basically macro animals eating these plants appears to be more common in these non-grassy areas because they have easy access to the seed heads basically of these plants. So you can imagine it's much easier for a deer to graze on this like a small field of Listemia and Plantago than it is to dig its head deep into the bottom of a patch of Avena and try to get anything out of there. The other thing though that I have noticed, which unfortunately I haven't been able to quantify in this experiment because it wasn't really designed to do that, is that there's this interesting thing that I've noticed a couple of times where when we have these tall grasses and it creates this shady and slightly moist environment, it seems to sort of facilitate herbivory by insects, like I imagine probably like earwigs and stuff. So I don't know too much about what's going on there or what the mechanism is, but there do seem to be different predation pressures perhaps in these different environments, which is an interesting thing.
Thank you for that. So we have time for just a few more questions. So this comes from Dennis Beebe, and he was asking about invasive grasses. Are invasive grasses more of a competition in a grassland than they are in hummocks? So it seems like there would be fewer invasive grasses in hummocks.
Indeed, yeah, that's definitely true. So most of these invasive grasses are going to be very rare, if not present at all, in the more stressful, abiotically stressful hummock environments. And it could be argued that that's probably one of the reasons that there's such a large discrepancy between what's driving the differential effects of competitors on these species, which basically just reinforces this idea that these native grasses, these invasive grasses in this sort of grassland matrix are really strongly negatively impacting native species that could be there, but they also don't occur in the hummocks as much, which sort of creates these spatial refugia for the native species. And in retrospect, I wish I had studied, I wish I had included in this study some invasive species as well, because it would have been really interesting to elucidate why, let's say, Avena doesn't occur as much in the hummocks. Is it environment? Is it that it can't survive early competition? I'm not really sure. Maybe some people have studied this as well, but that would have been an interesting thing to study in retrospect.
Well, thanks so much, Kenji. We're out of time here. This was a fascinating talk. We will be posting this on Vimeo. I'll be sharing the link for those who are here that want to watch it again, or for those who have missed the talk. There's a lot of really great implications for grassland conservation here, and so really appreciated that. And Thanks to everyone for joining us and asking great questions. Enjoy the rest of your day.
Thank you.
Bye-bye.